If you have ever found yourself scratching your head over topics like genetics or archaeogenetics, or maybe you've come across terms like haplogroups, mitochondrial DNA, or autosomal markers and wondered what on earth they mean, you're definitely not alone. Archaeogenetics can seem pretty complex at first, but don't worry, by the end of this video, you will have a solid grasp on what it is, how it works, and why it is so fascinating. So let us start with the basics. What is archaeogenetics? At its core, archaeogenetics is the study of ancient human DNA. This field helps scientists unlock the genetic secrets of our ancestors by examining preserved DNA found in human remains some thousands of years old. Through this, we can learn about human migration, about evolution and lifestyles from long before written history. But how do we even extract DNA from ancient remains? Well, over time, the DNA breaks down, but scientists have developed advanced techniques to recover and analyze even the tiny fragments of ancient genetic material. And that is how we basically extract DNA. Alright, so let us start with something called a haplogroup. What is a haplogroup? Haplogroups are special types of DNA that help us trace our ancestry in a straight line, one from the father's side and the other from the mother's. Each haplogroup traces back to a common ancestor, someone who lived thousands or even tens of thousands of years ago and passed on a mutation in their DNA. Now, what are mutations? I want you to imagine right now a long line of people passing a secret message from father to son. At first, the message stays the same. But one day, someone makes a small spelling mistake. Maybe they change one letter, and that little change stays in the message forever, and everyone after him copies the new version. Now, there are two versions of the message, the original and the one with the small change. The small change would be a mutation, and the group of people who carry it are part of a new haplogroup. Every time a new mistake happens in the message, a new group forms. Over time, these small changes help the scientists to see who is related and where their ancestors went. These haplogroups are passed down in two main ways: through the Y chromosome, which the fathers pass only to their sons, and through the mitochondrial DNA, which the mothers pass to all their children. Now let us focus on the Y-DNA. Y-DNA is like that secret message that only goes from the father to son. Every man gets it from his dad, who got it from his dad, and so on. And there are no females in this message chain. And just like I said earlier, when a little spelling mistake happens in the message, it creates a new version. That version is what we call a haplogroup. So Y-DNA haplogroups are basically the different versions of the father to son message, each one showing a new branch in the big human family tree. And by looking at which version you have, we can figure out which part of the tree your male ancestors came from. The oldest Y-DNA haplogroup is called haplogroup A. This haplogroup is found mainly in certain populations in Africa and is considered the most ancient branch of the Y chromosomal tree. When we trace all Y-DNA haplogroups back in time, they converge to one man. Scientists call him Y-chromosomal Adam. He wasn't the only man alive in his time, but he is the most recent common male ancestor from whom all men today have inherited their Y chromosome. From his lineage, different mutations happened, and these mutations created various Y-DNA haplogroups we see in men today. Now that we understand what Y-DNA haplogroups are, let us talk about subclades. A subclade is simply a smaller, more specific branch within a larger haplogroup. It is very similar to a haplogroup, but haplogroups are just more ancient and the mutations are more recent. Think of a haplogroup as a big branch on a tree, and the subclades as the smaller branches and twigs that grow from it. With time, new mutations appear in the Y chromosome. Each new mutation marks the start of a new subclade. This makes the haplogroup tree more detailed, which helps us to trace a paternal lineage with greater precision. We also use two different kinds of terms here. The first one being upstream subclades, the second one downstream subclades. Upstream subclades are the older, broader branches. They are shared by a large number of people. Downstream subclades are the newer, narrower branches shared by fewer people because they come from more recent mutations. I'll give an example. R1A is a major upstream haplogroup. A downstream branch of it is R1A Z93. A further downstream branch is R1A Z94. And even further, we have R1A L657. So if we have to say the upstream branch of R1A L657, we would say R1A Z94. And the upstream branch for R1A Z94 would be R1A Z93. And all of these subclades are downstream to the earliest mutation of R1A. And that's how we can trace which subclade is older and which subclade is a newer one. Every step you go down, it tells you more about where and when a haplogroup line split off in human history. The further downstream you go, the more specific the story becomes. Now let us talk about mtDNA, which stands for mitochondrial DNA. Unlike Y-DNA, mitochondrial DNA is passed down from mother to all her children. Both sons and daughters get it, but only daughters can pass it on to the next generation. Your mother gets it from her mother, and her mother gets it from her mother's mother, and it goes on like this. The mitochondrial DNA changes very slowly over time, which makes it useful for tracing deep maternal ancestry. Just like Y-DNA, mitochondrial DNA is also grouped into haplogroups. Examples include H, M, L, U, etc. Each haplogroup represents a branch of the maternal family line and can tell us about ancient migrations of women in human history. The oldest mitochondrial DNA haplogroups are found in Africa, and just like with Y-DNA, all human mitochondrial DNA lines trace back to one woman that the scientists call Mitochondrial Eve. She wasn't the only woman alive in her time, but she is the most recent common maternal ancestor of everyone alive today. Now we discuss autosomal DNA, the part of our DNA that makes up the majority of who we are. Before we begin, we should discuss chromosomes. We have a total of 23 pairs of chromosomes. The first 22 pairs are autosomes, while the 23rd pair consists of sex chromosomes, X and Y. So most of our DNA is autosomal DNA. The haplogroups make a small part of it, unlike Y-DNA or mitochondrial DNA, which only follow one single ancestral line. Autosomal DNA is a mixture from all your recent ancestors. From your parents, you get 50% each, 50% from your mother and 50% from your father. You get 25% each from your grandparents, and from your great-grandparents, you get about 12.5% each. With every generation, the share of DNA that you inherit from one ancestor becomes 50%, or you can divide it by 2. Go back 7 generations and you'll have 128 great-great-great-great-great-grandparents, and on average, each one contributes 1 by 128 of your DNA, which is about 0.78%. That's very tiny. So after 7 generations, an ancestor's direct autosomal influence is usually very minimal, or it may have vanished entirely, even though they still appear in your family tree. Autosomal DNA is also what affects many of our traits, things like hair color, skin tone, eye shape, height, lactose tolerance. All of these are influenced by your autosomal genes. But genetics is complex. Most traits are influenced by many genes together and not just one. I want to quickly discuss with you genotype and phenotype. Genotype is the actual genetic code in your DNA, and phenotype is how those genes are expressed physically. Phenotype is basically your face, the way you look, or your features, and genotype is basically how your DNA is structured. The main thing to remember is this, that autosomal DNA changes every generation, constantly mixing and reshuffling. It is what makes you unique, and it is what truly reflects your overall ancestry, appearance, and many traits, unlike the haplogroups. Now, that is something I really want to clear up, because I used to be confused about this myself when I first started learning about genetics. When you look at Y-DNA for a population, you might see something like 20% R1A or 30% J2. Many people think this means that a single person has 20% R1A and 30% J2, and maybe some other haplogroups also mixed in, like ingredients in a recipe. But that's not how it works. Those percentages are not about individuals. They are about the proportion of men in the tested group who have that haplogroup. For example, If a population shows 20% R1A, it means 20 out of every 100 men tested have the R1A Y-DNA haplogroup. The rest have other haplogroups. An individual man can have only one Y-DNA haplogroup, the one he inherited from his father's line. You cannot have some percentage of another haplogroup in your DNA. Back when I was new to this, I did not know the difference between haplogroups and autosomal DNA. I thought they were the same thing. So when I saw high percentage of R1A in a group, I assumed it meant a person had that much R1A in his DNA and other haplogroups in other percentages. But yes, Y-DNA does not work that way. This is why it is so important to understand the difference between Y-DNA and mtDNA and autosomal DNA. Haplogroups trace a single line of ancestry. Autosomal is what shows your admixture. Let me explain with a clear example to help you understand how haplogroups and autosomal DNA behave differently. Imagine a Ukrainian man who carries the Y-DNA haplogroup R1A. For whatever reason, he moves into sub-Saharan Africa and decides to stay there permanently. He marries a local woman and they have children. Now their children will be half European and half sub-Saharan African in terms of their autosomal DNA. The sons will carry their father's Y-DNA haplogroup R1A. I'm picking R1A for this example because a lot of Ukrainians have R1A. Alright, so the sons will carry their father's Y-DNA haplogroup R1A, but their autosomal DNA will be a mix of both European and sub-Saharan African. As the male children grow up and continue to marry local women, the European autosomal DNA portion will keep getting smaller with each generation because of constant mixing with the local gene pool. After about 7 generations of this mixing, the European autosomal DNA will be diluted so much that it might disappear completely from the descendants' DNA tests. But here is the interesting part. Even after all this mixing and autosomal dilution, the Y-DNA haplogroup R1A will still be passed down unchanged along the male line. It may have some mutations which will lead to different subclades being formed, but his descendants will still carry R1A. And because the European ancestry is barely present in the autosomal, this means that these descendants may look nothing like their Ukrainian ancestor anymore. Their physical traits and overall ancestry will mostly reflect the local population. This shows how haplogroups can survive long after the autosomal DNA signal fades away, and how Y-DNA can be helpful for tracing direct paternal lineage going back thousands of years. If you want to see more videos like this, do not forget to subscribe to the channel, and I will see you in the next video.